So welcome you back, my dear students. Hope you are very well at home because during this pandemic situation of COVID-19, it's very difficult. Well, we are continuing. We are talking about the DNA vectors. I hope you have already watched the part 1 video where I have highlighted the basic features of a DNA molecular vector. And now we are talking about the several examples of DNA vectors, that is maybe a expression vector, maybe a cloning vector. Okay, so in continuation with the previous video, the thing is PUC8 is the one more popular vector is the cloning vector. Okay, so is actually derivative of PBR322. You have already studied of PBR322. I have already told you in several times in the class. PBR322 is the most commonly used and popular DNA molecular vector. Plasmid vector is available in bacteria. And the basic features of this vector is the total size is 2686 BP, means the total genome size. Okay, total genome size is 2686 BP. So that means it's a very small size, and lag Z gene is also present. Lag Z gene, you may not know. Lag Z is the lactose synthesis gene, is the bacterial operon. In the ampicillin resistant gene, in the ORI means origin of replication site. So lag Z gene means here you can cut using restriction enzyme and you can insert your interest or target DNA and you can clone in the host cell. So next is the phage. Phage is also a very good common cloning vector, cloning large DNA fragments. That means in phage, you can clone a large size of DNA molecule or large size of gene. Suppose you are working with 100 base pair genes, so you can use PBR, PUC, these are the vectors. But when your gene size is more than 2 kb, 2000 base pair, more than 2 kb, so then only you can select lambda phage M13 or phage bacteria as a vector when you can clone the huge size of gene. It's a linear phase molecule. Linear means not the circular, it's a linear. Okay, used in storage of recombinant DNA and commonly used. This is the example of lambda phage M13 and phage bacteria, or the example of phage vector. Bacteriophage vector is here. I have already just told lambda phage and M13 are very commonly used in E. coli phages. Its genome size, see here, 48,502 BP. So it's the large genome size, lambda phage. Lambda phage is the large genome size. That's why in the large genome size you can also clone the large gene. Well, so high transformation efficiency and 1,000 times more efficient than the plasmid vector. Plasmid vector means PBR322, PUC8. These are the plasmid vector, small circular bacterial vector. What is the lambda phage or M13? These are the biggest size of vector, and it is the linear DNA, and you can cut in between the DNA and you can clone the interest gene. So here you can see is the M13 vector. M13 vector, it has the lag operon promoter. Lag operon promoter is the lactose synthesis gene. Polylinker. Polylinker means here you can insert multiple copies of genes, and it is the M13 genome. Is the total genome is near about 5,000 base pair. Here is the lag gene, here is the another gene, selective marker gene, and here is the polylinker, means here you can insert your gene. And the whole thing, the recombinant molecule, recombinant because this is the actual genome, vector genome, and these are your gene of interest you have inserted. Okay, then the total genome is termed as the recombinant because it's the recombination of the genomes, two different genomes. So that genome you can clone inside the host cell. Now is the thing is hybrid vector. Hybrid vector is also a very useful vector, and is the component from both plasmid and phase chromosome. Hybrid means the combination of two different things. So here the plasmid vector and the phase vector, the low genome size and the high genome size, from their chromosome this hybrid vector is developed, and developed in 1978 by Barbara Horn and John Collins. These are the scientists, and its size C 30 to 40 kb. So in between the plasmid and phase chromosome, because the plasmid size is something about below 3000 base pair, and phase is 50,000. So in between, because this is the hybrid vector, in between this size is 30 to 40,000 base pair, is the kb is the base pair. Okay, or they have the origin of replication site, cooling site smaller than plasmid, and the example is cosmid. Okay, though, so if I ask the in the exam also, if they ask name one hybrid vector, molecular cloning vector, your answer is cosmid. Okay, then the cosmid is the detail about the cosmid. It is also have the cos site. These are the very detail you may learn during your postgraduate degree. So these are the detailed things, and structure of the cosmid is origin of replication site. ORI site. ORI means origin of replication. Means once you have inserted your gene must be multiplied. Okay, so it will be multiplied when replication will be started. So origin of replication, ori, this origin means, ori part is means origin of replication. And they have the restriction site. Restriction site means where the restriction enzymes endonuclease can cut, okay, and insert the foreign DNA molecule. Okay, so this is a basic feature of a cosmid vector. Is the hybrid vector is the restriction site. These are the restriction. Here is the ori site. Okay, antibiotic resistant genes is the selectable marker gene. Okay, so this is a typical structure of cosmid vector. Now, artificial chromosome. We are using the DNA molecule directly. We can use the artificial chromosome, not the natural chromosome. Here, mind it, it's the artificial chromosome. Artificial chromosome can be used as a vector. It is really linear or maybe circular. Okay, one or two copies per cell. It may be present in a cell. One copy may be present or two copy. Both are possible. And linear, it may be a linear means the straight line like, or maybe circular is a round shape. Okay. So different types of artificial chromosome, like bacterial artificial chromosome is called BAC, BAC, yeast artificial chromosome YAC. Okay. POI derived artificial chromosome pack, mammalian artificial chromosome MAC, even human artificial chromosome. See the interesting, human artificial chromosome also can be used in cloning for gene hack. And YAC is the yeast artificial chromosome in yeast. Saccharomyces cerevisiae is the yeast. BAC or PAC is the bacterial cell, and mammalian and human cell is the MAC and HAC. Here, mammalian and human. Okay, so these are the example. So bacterial artificial chromosome is the some features. The example is PBAC1080L is the example, cloning of large region of eukaryotic genome. Eukaryotic because you can work with eukaryotic genome. Origin of replication site is also there, and F factor is the sex pili factor, is a very difficult thing for conjugation or transformation in the bacterial gene transfer event. Bacterial artificial chromosome vectors are, these are the examples and used in analysis of genome, host in these are the features you can write down in your notebook and you can memorize for your exam. And each artificial chromosome also having some features, linear vector, and its size is this one. It may be circular. Here is the example. It contains ARS sequence and centromeric function. Selectable marker is very common. Selectable marker because when you have cloned a gene into the bacterial cell, you need to multiply the colony on a medium. So there in the medium you have to give the antibiotic resistant. These are the selectable markers. Okay. So here is the typical picture of a gene cloning. You can see there is the overall thing. Suppose in a bacteria, bacterial genome is there, but bacterial circular DNA is the plasmid. Suppose this is a PBR322 or PUC8. So this vector you just extract it through bacterial cell lysis using a buffer. Then the vector has to be cut using restriction enzyme, so that once it is cut you can insert your foreign gene of interest. Then see here it has been inserted. So this is the recombinant DNA molecule. Now transformation, this recombinant molecule again you are transforming into the host cell. Okay. So host cell, when it will replicate from 1 to it will make 2, from 2 to it will make 4. So likewise the recombinant molecule will be also multiplied. Okay. So in case of bacteria, in case of virus also, you can see there are linear DNA. Here is the circular DNA, there is a linear DNA, viral DNA, and viral DNA also cut by restriction enzyme, then joined with the ligase and the recombinant molecule. This total molecule can be inserted into the host cell, and host cell can be multiplied on lysis. Then we'll make the multiple copies. So this is the overall thing about the DNA molecular vector. Hope you can understand. And if you have any doubt, I give you one suggestion. You need to watch this video several times, pause it, write down the whole things in your notebook, then you will be able to understand the whole things. Thank you for watching.